Hello everyone, welcome to the concept and fact of life sciences. Today in this video, we are going to discuss about the amino acids. Amino acids, they are L-amino acid. Naturally occurring amino acids, they are L-amino acids because their structure resembles to the L form of glyceraldehyde. In our previous video, we already stated that the naturally occurring sugars, they are the D sugars because their structure resembles to the D form of glyceraldehyde. So the naturally occurring amino acids, they are L-amino acids. Other functions of amino acid includes number one, standard amino acid. Amino acid which are added during the process of translation are called as standard amino acid. There are 20 standard amino acids. Recently, two new amino acids has been discovered which are added during the process of translation. These includes the salicylic and pyrrolysine. Salicylic, it is found in case of both prokaryotes and eukaryotes, while the pyrrolysine found only in the case of prokaryotes. So there are 20 plus 2, 22 standard amino acids. Amino acids, they also act as the neurotransmitter. Neurotransmitters, they are of two types. There are neurosuppressor type of neurotransmitters. There are neuroexcitator type of neurotransmitters. If we talk about the neurosuppressor, there are examples of glycine as well as there is GABA. GABA is gamma amino butyric acid, which is generally produced by the beta cells of pancreas, and it will act like a neurosuppressor. Similarly, there are neuroexcitator type of neurotransmitter. These includes the aspartates or aspartic acid and glutamate or glutamic acid. So there is aspartic acid and glutamic acid which will act as a neuroexcitator. Now the amino acid, they will also act as the intermediates of metabolic pathway. For example, there is an example of citrulline, there is ornithine, there is arginosuccinate. These all are the example, or we can say these are the intermediates of pathway which is called as the urea cycle. So these are also the amino acid, but these are not the standard amino acid. These are the intermediates of metabolic pathways. So we can say there are more than 500 types of amino acid known, out of which only 22 are standard, which are added during the process of translation, जो प्रोटीन संश्लेषण में काम आते हैं. Next, characteristic feature, alpha amino acids. Amino acids, they are known as, or we can say the standard amino acid, these are the alpha amino acids because the functional group remain attached to the alpha carbon. So it is the basic structure of an amino acid. It is the framework structure, while there is a R chain which determines the type of amino acid. So the functional group remain attached to the alpha carbon. So the standard amino acid, they are alpha amino acids. Except, there is an exception, or we can say in biological system, exceptions are always there. Except an amino acid which is known as proline. The proline is known as the alpha amino acid. It is a alpha amino acid. Why this is called as alpha amino acid? Because here the amino group directly forms and bond with the carbon, double bond with the carbon, sorry, and there is a formation of a chain structure, sorry, ring structure. So this is called as the alpha amino acid. While all other amino acid or all other standard amino acid except the proline, they are alpha amino acid, while the proline is alpha amino acid. Next is optically active. All the amino acids or all the standard amino acids, they are optically active, except, again there is an exception, and the exception is the glycine. Means if a molecule is having a chiral carbon, if it is having a chiral carbon means if all the valency of this carbon it is satisfied by different groups, then it is known as the chiral carbon. Here we can see if here it is a H atom, amino group and carboxylic group. If here we are having an H, which is found in case of glycine, the R is equals to H. So if we replace the R with H, there are two hydrogen and the carbon is now not a chiral carbon. So the molecule which is having the chiral carbon, it is able to rotate the plane of plane polarized light, either levorotatory or the dextrorotatory. So except the glycine, all other standard amino acids, they have chiral carbon and have ability to rotate the plane of plane polarized light and thus known as optically active. Glycine is not optically active. Next is the classification of amino acids. Amino acids, they can be classified as hydrophobic aliphatic, in which we are going to study the glycine, alanine, valine, leucine and isoleucine. There is alcohol containing amino acid, in which we are going to study the serine and serine and threonine. Sulfur containing, there are cysteine and methionine. There are basic amino acid which includes the lysine, arginine and histidine. There is aromatic amino acids which includes phenylalanine, tyrosine and tryptophan, and there is another group which is known as dicarboxylic acid that includes the aspartic acid and glutamic acid, which also, as we already stated, act as a neuroexcitator. There is dicarboxylic acid in the form of glutamic acid and aspartic acid, or we also have their amides in the form of glutamine and asparagine. So there are 20 standard amino acids which are divided into these 6 groups. Except this, these are 5, 2, 7, 2, 9, 3, 12, 12 and 3, 15 and 4, 19. These are 19 standard amino acids, and we also have one that is known as the alpha amino acids. Next, we will discuss one by one. First, we are taking the hydrophobic aliphatic group that includes the glycine. Glycine, it is abbreviated as GLY and it is denoted by the alphabet G. As we already stated, here the R is equals to H. If we put the value of R equals to H, it will become glycine. Now, the alpha carbon or the carbon, now it is not a chiral carbon. So, it is optically inactive, we can say, glycine. Next, it is smallest known amino acid. It is smallest known amino acid which do not fit in any regular structure. That's why it is also known as the alpha helix breaker, or we can say it do not allows the formation of stable alpha helix, that is the secondary structure of proteins. These are neither hydrophobic nor these are hydrophilic. So ना तो ये हाइड्रोफोबिक है ना ये हाइड्रोफिलिक है. So these are generally used to estimate the hydrophilicity and phobicity of other molecules or amino acids. If the glycine it get methylated, अगर glycine का आप methylation कर देंगे, methyl group ऐड कर देंगे, then it is known as the glycine betaine. It is known as glycine betaine. Now this glycine betaine, it is known as a compatible osmolyte. It is a compatible osmolyte. Compatible osmolytes are those which helps the plant to fight or to we can say survive under the stressful condition. So इसके बारे में आगे अपन बात करेंगे प्लांट फिजियोलॉजी के अंदर. So if the glycine it get methylated, it is known as glycine betaine, and this glycine betaine is a compatible osmolyte. Next is the valine. Valine, it is abbreviated as VAL, and it is denoted by the alphabet V. The R chain of valine, it contains this. Simply we can put the value of R, that is CH3 and CH3. Now this molecule, it is having a one chiral carbon, like the valency of this carbon, it is having H, NH2COH and this group. So four different valencies satisfied by different type of groups. So we can say it is having a chiral carbon and it is optically active. Now these property of these valine, that they helps in the muscle growth and tissue repair, as well as they supports the nervous system. Next type of amino acid is the alanine. Alanine, it is abbreviated as ALA and denoted by the alphabet A. Here the value of R is equals to CH3. We can simply put the value of R here and it will become the alanine like this. So it is derived from the pyruvate, and we can say the pyruvate will act as the precursor for most of the amino acids. Next is the leucine. Leucine amino acid is abbreviated as LEU and it is denoted by the alphabet L. Here the value of R is this. Next type of amino acid is the isoleucine. Isoleucine, it is abbreviated as ILU and it is denoted by the alphabet I. Here the value of R is this. If you put the value of R in this framework structure, then it will become CH, CH2, CH3, and we are also having this CH3. Now also we can also written it like this. Okay. Now here there are two chiral carbon, this chiral carbon as well as this chiral carbon. Now it is having four valencies, H, NH2COH and this particular group. So it is a chiral carbon. While this C, it is having one H, one this entire group, CH3 and CH2, CH3. So the isoleucine is having two chiral carbon. ये आपको याद रखना होगा. It is having two chiral carbon. Next category is the alcohol containing amino acid. The amino acid which are having OH group in their side chain, they are known as the alcohol containing amino acid. There are two alcohol containing amino acid. One is serine, other is known as the threonine. Serine, it is abbreviated as SER, denoted by the alphabet S, while the threonine is abbreviated as THR and denoted by the alphabet T. Here the value of R in case of serine, it is CH2OH. Simply we can put the value of R, that is CH2OH, and it will become the amino acid serine. Now serine, it is the site for O-linked glycosylation. Simply glycosylation, what is glycosylation? It is the simply the addition of sugar to the molecule or to the protein, that it is called as glycosylation. Now this glycosylation, it is of two types. One is called as the O-linked glycosylation. इसके बारे में आगे अपन बात करेंगे जब अपन सेल ऑर्गेनल्स की बात करेंगे. And other is known as the N-linked type of glycosylation. Now the O-linked glycosylation, it generally occurs in the Golgi body, while the N-linked glycosylation, it occurs in the endoplasmic reticulum. Now this serine, it provides the site for O-linked glycosylation. The sugars can be added on this molecule. So this involve in the formation of glycosidic bond, or we can say this involve in the process of O-linked glycosylation. It also remains present at the active sites of enzyme where the catalysis occurs. Next type of amino acid, it is the threonine. Here the value of R is this. If you put this in this framework structure, it will become like this: C H OH CH3. Now again, like isoleucine, the threonine, it also have two chiral carbon. We can see over here. So ये चीज याद रखनी है आपको. The isoleucine and the threonine, they are having two chiral carbons. Next are the sulfur containing amino acids. The amino acids which have sulfur in their side chain, they are known as sulfur containing amino acid. Again, there are two types of amino acid which have sulfur in their side chain. One is known as the cysteine, other is known as the methionine. Cysteine, it is abbreviated as CYS and denoted by the alphabet C. Its side chain contains the CH2SH group. We can see the structure of cysteine over here. It is the basic framework structure which is having CH2SH group as a side chain or R chain, we can say. And it is another molecule of cysteine. ये अभी क्यों बनाया है उसको भी समझाते हैं आपको. So if the two cysteine, they are in front of each other under the oxidizing environment that is provided by the endoplasmic reticulum, then they have tendency to form a disulfide bond. And they have tendency to form a disulfide bond, and after formation of disulfide, this is now known as the cysteine. So there is cysteine and there is cysteine, and these cysteine, they absorbs the UV radiation of 280 nm. Generally, the UV radiation is absorbed by the aromatic amino acid, but if the cysteine, it is having a disulfide bond, it is a disulfide bond. If a cysteine is having a disulfide bond, then it is called as cysteine, and which is now able to absorb the UV radiation of 280 nm. It occurs under the oxidizing environment of endoplasmic reticulum, and as we know that the pathway of secretory protein, it passes through the endoplasmic reticulum. So these disulfide bonds, they generally found in case of secretory protein and provides extra stability. There are two type of proteins, one is secretory, other is cytosolic proteins. Secretory proteins का जो पैसेज है आगे अपन बात करेंगे, इनकी जो टारगेटिंग होती है वो एंडोप्लाज्मिक रेटिकुलम के थ्रू होकर के होती है, इसलिए वहां पे ये डाइसल्फाइड बॉन्ड फॉर्मेशन हो सकता है, और अगर होता है तो अपन कहते हैं इट प्रोवाइड्स फर्दर स्टेबिलिटीज टू द प्रोटीन्स. नेक्स्ट अपन बात करते हैं मिथियोनिन के बारे में. मिथियोनिन, इट इज एब्रिविएटेड एज MET and denoted by the alphabet M. Here the side chain, it contains this structure. Methionine, as you all know, that it is added onto the first position of proteins, like the start codon. The start codon is AUG. This is the start codon. This start codon, it encodes for the methionine. The start codon, it encodes for the methionine, but it generally remain present in least amount within the proteins. ऐसा क्यों होता है? ऐसा इसलिए होता है क्योंकि एक तो जो रिडंडेंसी और डीजेनरेसी होती है, मींस आगे अपन बात करेंगे जब हम मॉलिक्यूलर बायोलॉजी पढ़ेंगे, देयर इज मोर देन वन कोडॉन फॉर ए पर्टिकुलर अमाइनो एसिड, but in case of methionine there is only a single codon, and that is also a start codon, but still it remain present in least amount. Why? Because these first positions, they generally contains the targeting sequences. Targeting sequences means these are the signal peptides which helps the particular protein to transport towards a particular cell organelle, and after reaching to that particular site, these targeting sequences, they will be cleaved by the enzyme signal peptidases. Therefore there is a presence or there is a least presence of this amino acid within the polypeptide. Now this is the structure of methionine. लाइक यहां तक आपका फ्रेमवर्क स्ट्रक्चर, और इसके आगे आपका जो आपने ग्रुप यहां पे बनाया है वो आपने ऐड कर दिया. सो दिस इज द स्ट्रक्चर ऑफ methionine. If this methionine, it get the addition of adenosyl groups, we can say if there is a addition of adenosine. Adenosine, it is a nucleoside which includes the adenine. Adenine is a nitrogenous base plus a pentose sugar, then it is called as adenosine. If there is an addition of this adenosine to the methionine, then it is called as the S-adenosyl methionine. If there is an addition of this to this molecule, then it is now known as the S-adenosyl methionine. Now this S-adenosyl methionine, it will act as a methyl donor. Okay, methyl donor, and also helps in the process of DNA methylation. आगे बात करेंगे अपन इसके बारे में. So if there is an addition of adenosine to the methionine, it is now known as the S-adenosyl methionine, which can be act as a methyl donor, or which helps in the transfer of methyl group to the DNA, or which helps in the process of DNA methylation. So this is all about the sulfur containing amino acid. Next अपन बात करते हैं, there are basic amino acids. Now within this basic amino acid, there are three types of amino acid. One is known as the lysine, second is known as arginine, and third is known as the histidine. बात करते हैं तीनों के बारे में. Lysine, it is abbreviated as LYS, क्योंकि आपने ल्यूसिन के लिए L काम में ले लिया है, तो आप L अल्फाबेट काम नहीं ले सकते. So on the basis of pronunciation, lysine, it is denoted by the alphabet K. The side chain of lysine, it contains this group. So it is a basic amino acid. Now what is the function, or what are the function of this lysine? That it provides basicity. So it is a basic amino acid, and we know that histones, protein, they are made up of basic amino acids, and they are positively charged. Due to the presence of this basic amino acid, the histone is positively charged, and it can interact with the DNA. जैसे कि अपन ने पहले बॉन्डिंग में बात की थी, the DNA, it is having partial negative charge, while the histone, they are having partial positive charge, and there is a ionic interaction between the DNA and histone. So we can say the basicity of histones, they are due to the presence of lysine as well as the arginine. The lysine and arginine, they provide basicity to the histone protein. Now next is the nuclear localization signals. जैसा कि अपन ने पहले भी बात की थी प्रोटीन टारगेटिंग की, है ना, प्रोटीन टारगेटिंग. Now the protein which is to be targeted towards the nucleus, they are having the nuclear localization signal, or we can say these are having the NLS. Now this NLS, it is also made up of repeated units of lysine and arginine. Next is the ubiquitination. Ubiquitination of protein means this the degradation of protein simply. Now the ubiquitin basically the ubiquitin, it is a protein of around 38 amino acids. If the ubiquitin is added to the protein is going to be degraded. Now, there are two types of ubiquitination process. One is called as the polyubiquitination and other is known as the monoubiquitination. If there is an addition of a single ubiquitin, then it is called as monoubiquitination. If there is an addition of multiple ubiquitin, it is known as the polyubiquitination. So if there is an addition of ubiquitin to the protein, then the protein is going to be degrade. Where is to be degrade? It depends upon the how much amount, or we can say how much units of ubiquitin are added to that particular protein. If there is addition of single ubiquitin, then the degradation site, it will be lysosome, and if there is addition of multiple, then the degradation site will be proteasome, which is the part of साइटोप्लाज्म. तो साइटोप्लाज्म कहो या प्रोटियासोम कहो, दोनों ही सही है. So this ubiquitin, it is generally added to the lysine amino acid. जो प्रोटीन में लाइसिन अमाइनो एसिड है उसी के ऊपर यूबिक्यूटिन ऐड किया जाता है. That's why we are stating that it also involved in the ubiquitination of proteins. आगे भी इसके बारे में बात करेंगे अपन जब प्रोटीन्स पढ़ेंगे. Next is the arginine. Arginine, again, it is a basic amino acid. It is abbreviated as ARG and denoted by the alphabet R. Because alanine में आप एक आम ले चुके हैं, तो arginine यहाँ डिनोट कर रहे हैं अपन अल्फाबेट R से. साइड चेन के अंदर आपके ये ग्रुप होता है, एंड दिस साइड चेन it is having a name. That's why it is important. It is a guanidine side chain, and it is more basic than the lysine. तो एक तो चीज याद रखनी है आपको कि it is having the guanidine side chain and it is more basic. It provides the basicity to the histone. It found in case of the NLS, and it is more basic as compared to the lysine. Next is the histidine, abbreviated as HIS and denoted by the alphabet H. Now, it is again having a name, side chain. The side chain or the R chain of histidine, it includes a imidazole side chain. This imidazole side chain or this histidine is very much important because the side chain of this histidine, which is known as imidazole side chain, it is having the pKa value around the physiological pH. Physiological pH is 7 and it is having the pKa value of 6.9. So pKa आगे बात करेंगे अपन. pKa is the pH on which the acid it remains half, or acid or any other molecule, it remains half associated and half dissociated. So if it remains as half associated and half dissociated, it can act as a buffering agent. So hemoglobin active site पर जो histidine है, it also acts as a buffering agent. कैसे अपन आगे बात करेंगे. इंपॉर्टेंट चीज है कि इसकी जो pKa साइड चेन की pKa वैल्यू होती है, pKa is the pH on which the acid or molecule is half associated and half dissociated. वो physiological pH के अराउंड है, इसलिए they can act as a buffering agent. They are also the precursor of histamines. They are involved in the process of metal ion coordination. बात करते हैं इसके बारे में. पहले बात करते हैं histidine. In case of the hemoglobin, it is acting as a buffering agent. Hemoglobin, as we all know, that it is a tetramer having 2 alpha and 2 beta subunit. All of these subunit, that is alpha 1, alpha 2, beta 1, beta 2, they are having a central heme atom, and it also contains the Fe in their center. Okay. Now there are 38 residues of histidine per tetramer that regulates the blood pH. कैसे करेंगे, कैसे रेगुलेट करते हैं, बात करते हैं। इमिडाजोल साइड चेन की बात की थी अपने ये कहा था कि इसकी जो pKa वैल्यू है दैट इज अराउंड द फिजियोलॉजिकल pH, इसलिए ये एक बफरिंग एजेंट के रूप में काम कर सकता है। नाउ द इमिडाजोल साइड चेन इट कैन remain in two forms. One is known as the proteinated form, other is known as the deprotonated form. Proteinated, if it remains at the pH lower than the 6.9, then there is a acidic environment, we can say, or we can say the amount of H plus ion it increases. Under that condition, it will take the H plus ion from the solution and resist the change in pH. Similarly, at the higher pHs where there is a lower amount of H plus ion, it donates it H plus ion and resists the change in pH. So we can say at the lower pH where there is a higher amount of H plus ion, as we all know that the pH is the negatively proportional to the H plus ion concentration. So if the pH is decreasing, H plus ion concentration is increasing. Under that particular condition, the solution it is having more H plus ion, so the deprotonated form it will take up the H plus ion when will convert into this particular form. And if the pH it increases, there is less H plus ion, there is more OH ion. Under that particular condition, it will leave the H plus ion and will convert into this particular form. So there is deprotonated and protonated forms which resist the change in pH. That's why it can act as a buffering agent. Now, each active site of hemoglobin, it is having two histidines. One is called as the proximal histidine, other is called as the distal histidine. जो proximal histidine है, it directly binds to the Fe, it holds the Fe and it increases the affinity of hemoglobin towards the oxygen. While जो distal position पे आपका histidine है, it will not directly binds to the Fe, but it forms the hydrogen bond with the bound oxygen and stabilize the structure and also increases the affinity of hemoglobin towards the oxygen. ये affinity of hemoglobin increase कर देता है, या oxygen binding को ये enhance कर देता है, we can say. So this is all about the histidine. Next is the aromatic amino acid. They are of three types: phenylalanine, tyrosine, and tryptophan. Phenylalanine, it is abbreviated as PHE, denoted by the alphabet F. Tyrosine by TYR, denoted by Y. P, क्योंकि आप प्रोलिन को दोगे तो यहाँ पे आपको phenylalanine F अल्फाबेट से डिनोट कर रहे हो. टायरोसिन T आप पहले थ्रियोनिन को दे चुके हो, इसलिए Y से आप डिनोट कर रहे हो. ये स्ट्रक्चर R जो साइड चेन होती है उसके अंदर R इक्वल्स टू CH2 और एक बेंजीन रिंग होती है इसके अंदर. अगर if there is a presence of enzyme, this is known as phenylalanine hydroxylase. If there is a hydroxylation of this phenylalanine, it will convert into the tyrosine. Means simply there is an addition of an OH group. Then it is known as tyrosine. But if this enzyme phenylalanine hydroxylase, if it gets mutated, in that particular condition, this phenylalanine, instead of converting into the tyrosine, it will convert into the phenyl pyruvate. And this phenyl pyruvate, it is a ketone body that leads to a disease which is known as phenylketonuria. So there is a phenylketonuria if there is an absence of enzyme which can convert the phenylalanine to the tyrosine. Similarly, another type of aromatic amino acid is known as the tryptophan. Tryptophan, it is abbreviated as TRP and denoted by the alphabet W because its structure resembles with the alphabet W. Now it is a precursor of indole acetic acid, which is a plant hormone, auxin or indole acetic acid. It is a naturally occurring auxin. It is having the maximum absorption at 280 nm. Means as we already stated that the aromatic amino acid, they are able to absorb the UV radiation of 280 nm, or we can say that the protein can absorb the UV radiation due to the presence of these aromatic amino acid, and the maximum absorption at the 280 nm, it is by the amino acid tryptophan, and its side chain, it is having a indole side chain. So this is all about the aromatic amino acids. Next, it is alpha amino acid, and we already stated that it is proline, abbreviated as PRO, denoted by the alphabet P. स्ट्रक्चर के बारे में अपन पहले भी बात कर चुके हैं. Here the amine group, it is directly involved, or it binds to the carbon with a double bond, leading to the formation of a ring structure that is called as pyrrolidine. It is also known as the alpha helix breaker. It do not fits in the protein secondary structure. Protein secondary structure, it is having two type of structure, that is alpha helical and beta sheet structure. So जो alpha helix है उसका ब्रेकर कहलाता है क्योंकि उस रेगुलर स्ट्रक्चर में ये फिट नहीं हो सकता होता है, बिकॉज इट इज नॉट ए अल्फा अमाइनो एसिड, इट इज ए अल्फा इमिनो एसिड. सिमिलरली जनरली द प्रोटीन्स दे फॉर्म्स ट्रांसपेप्टाइड बॉन्ड्स, है ना. अमाइनो एसिड दे फॉर्म्स ए द पेप्टाइड बॉन्ड, इट इज द बॉन्ड व्हिच रिमेन, और कोवैलेंट बॉन्ड व्हिच रिमेन प्रेजेंट बिटवीन द अमाइनो एसिड एंड अमाइनो एसिड. It generally remains in a trans configuration naturally, but the proline, it forms cis and trans both type of peptide bond. Remaining all they forms trans peptide bonds, while the proline it forms both cis and trans peptide bond. Generally the proline they also forms we can say the trans peptide bond, but there is an enzyme which is called as prolyl cis trans isomerase, which converts these trans peptide bond into the cis peptide bond. So it is again an exception which is having both cis and trans peptide bonds, and it is known as alpha helix breaker, we can say. If the proline they remain present in a repeated form, like in polypeptide there is a repetition of proline, and proline, then it is known as the polyproline sequences. It is having a distinct type of alpha helical structure that is called as polyproline second. Similarly, it is also acting as a compatible osmolytes which helps the plant to stay under the abiotic stress conditions. Next is the dicarboxylic acids and their amides. First we are taking the example of aspartic acid, abbreviated as ASP, denoted by the D, side chain having a acidic group CH2COOH. It is derived from the oxaloacetic acid of TCA cycle or the Krebs cycle, tricarboxylic acid cycle. The amide of aspartic acid is aspargine, abbreviated as ASN and denoted by the alphabet N, aspargine. R chain, it contains this group. So it is, we can say, amides. It is the site for N-linked glycosylation. As we already stated, there are two type of glycosylation, O-linked glycosylation and N-linked glycosylation. The site for O-linked glycosylation अपन ने बात की थी कि that is the serine, while the site for the N-linked glycosylation, it is the aspargine. Next is the glutamic acid. It is abbreviated as GLU and denoted by the alphabet E. Side chain contain an acidic group. It is derived from the alpha ketoglutarate 5-carbon intermediate of TCA cycle or the Krebs cycle. The amide of this Glutamic acid, it is the glutamine, abbreviated as GLN, denoted by alphabet Q. Side chain contains this group, and it is important because glutamine is the transport form of ammonia from tissue to the liver. So this is all about the dicarboxylic acid and the amides. Next are the essential versus non-essential amino acid. Essential amino acids are those which cannot be synthesized or biosynthesized by our body. They are known as the essential amino acid, while the non-essential amino acids are those which are able to synthesize by the human body. So if we talk about the non-essential amino acid, they includes the histidine, H for the histidine. There is glycine, lysine. There is M for the methionine. There is phenylalanine. There is T for threonine. There is tryptophan, isoleucine, leucine, and val. So these are the essential amino acid which we required from outside source. Our body is not able to synthesize these amino acids. That's why they are essential. Remaining all standard amino acids, they are non-essential. Glucogenic and ketogenic amino acids, the amino acids which are able to convert into the glucose. Any amino acid which is able to convert into the glucose, it is known as the glucogenic amino acid. And if an amino acid converts into the ketone bodies, then it is called as the ketogenic amino acid. So there is glucogenic and there are ketogenic amino acid. Leucine और lysine को अगर छोड़ दें, except the leucine and lysine, all amino acid they are glucogenic. सारे के सारे जो 18 amino acids हैं, they are glucogenic, while the leucine and the lysine they are ketogenic. They first will convert into we can say the PC cycle intermediate, that is the acetyl coenzyme A, and forms ketone bodies. So there is glucogenic and ketogenic amino acids. Similarly, if we talk about the animal versus plant, in animals the amino acids are the source of energy, but in case of plants the amino acids are not the source of energy. Why? Because the plants they are having a specific type of TCA cycle or modified version of TCA cycle, that is called as glyoxylate cycle. Now, through this glyoxylate cycle, the plants have ability to convert the fatty acid into the glucose. Okay, so they do not require amino acid as a source of energy under the emergency condition. While in case of animal, there is no glyoxylate cycle. There is a special type of peroxisome which is called as glyoxisome, in which there is a glyoxylate cycle in case of plant, while the animal they are not having this particular type of structure. So there is a lack of this glyoxylate cycle. That's why the amino acids are the source of energy in case of animals. Next, the 21 and 22nd amino acid, they are newly discovered. Not newly, we can say the 22 amino acids है, 22nd amino acid ये भी 2002 में खोजा जा चुका था, तो अभी नया नहीं कह सकते, but these are additional to the our traditional 20 amino acid. One is known as the selenocysteine. It is denoted by U and abbreviated as SEC. It is the structural analog of cysteine. सिस्टीन का क्या स्ट्रक्चर था? अगर अमीनो एसिड की स्ट्रक्चर की बात करें अपन, CH and COH, it is the basic structure, and there is a cysteine that is having a sulfur group. Now this SH, if it is replaced by a selenium ion, then now it is known as the selenocysteine. So simply it is the structural analog of amino acid cysteine. It plays an important role, we can say, in enzyme glutathione peroxidase as well as the thyrodoxine reductase. Both of these enzymes, they are having very much importance in the biological system. So it is helpful or it is essential for the functioning of these two enzymes. क्या काम है आगे अपन बात करेंगे. This selenocysteine, it is encoded by the codon UGA. This UGA, it generally acts as a stop codon. But for selenocysteine, the codon is UGA. It can code for the selenocysteine through a process which is known as translational recoding. कैसे होती है बात करते हैं इसके बारे में. There is a specialized type of tRNA which can be charged with serine first. For the synthesization of selenocysteine, the TR, or we can say for the insertion of selenocysteine in the polypeptide, the tRNA, that specific type of, it is not the ordinary tRNA, it is different type of tRNA which is first charged with a serine. Now this serine tRNA, it is not, because it is on a different or it is on a specific type of tRNA, the elongation factor, it is not able to, elongation factor, it is involved in the process of translation and carry out the elongation of polypeptide chain. The elongation factor, it is not able to recognize that particular charged tRNA because it is different, it is not ordinary tRNA. Then there is an enzyme which is called as selenocysteine synthase. This selenocysteine synthase, it converts or it replaces this seryl group by a selenium. So it will become seryl tRNA. Now this tRNA, it can be recognized by the eukaryotic elongation factor 2 and can be added in the polypeptides. In case of, we can say the selenocysteine, it is found in case of both prokaryotes as well as in case of eukaryotes. तो यूकैरियोट्स में भी आपको देखने को मिलेगा सेलिनोसिस्टीन और प्रोकैरियोट्स में भी देखने को मिलेगा. In case of eukaryotes, beside this selenocysteine synthase, one more enzyme is required that is called as PSTK, that is phosphoseryl kinase. This is an enzyme which is also required in addition to selenocysteine synthase in case of we can say eukaryotic system. Next type of newly discovered amino acid is pyrrolysine. It is abbreviated as PYL and denoted by the alphabet O. It is found only in case of the prokaryotic system, found in case of archaebacteria and also in some bacteria. It is again encoded by a stop codon which is called as the UAG. For UAG to code for pyrrolysine, there should be a presence of the gene which is known as PYLT. These genes are necessary for the encodation of this pyrrolysine through a UAG codon. Now this is the structure of pyrrolysine, or we can say it is formed by the addition of two lysine molecules, that is known as the pyrrolysine. So this is all about the amino acid. Thank you.